Welcome back. I hope you weren't too overwhelmed with that. It's repeating a lot of the same material multiple times. So one of those explanations I hope will fit in your head. The other thing is, a lot of students ask me at this point, why am I learning this? I'm going to be a doctor. I don't care about photosynthesis. I get it. Well, I don't. I love photosynthesis. But I get it. It's not seeming very relevant. The thing is, one, all your food comes from the photosynthetic process. It's important to understand how energy flows. And we're going to have electron transport chains, ATP synthase. We're going to have very similar proteins involved in cell respiration. And you know that's something you have to learn for med school and your GREs and your professional exams. And basically to function as a professional scientist, you need to know that material. This is actually the easier way to explain it than just throwing you into cell respiration. So a couple good reasons to hold on to why you're learning this. And let's do the last bit here. This, I ask you to write down the formula at the end of the last lecture, or the video, the end of the last video. What are we missing here? We've used the water, we split water. We got rid of carbon dioxide, or we got rid of oxygen. So we did this, we oxidized water. We've taken those electrons and they have reduced other things. We've reduced NADPH, but we haven't reduced carbon dioxide. We haven't even seen carbon dioxide yet. We're going to use that ATP and the NADPH we just made to reduce carbon dioxide into sugar. But we have to do it in little baby steps, because if we try to just do this, carbon dioxide to sugar, there'll be so much energy lost as a part of that process, it's not going to be useful. We need to do it slowly and carefully by passing those electrons along slowly in order to drive this process as efficiently as we can. So here I've put this is my drawing. I think your drawing should look something like this. I tried drawing it all by hand and it was just a mess. So I just took the picture from the textbook and I laid it. This part would be on the thylakoid membrane. The hydrogens are building up inside the thylakoid lumen, here, boop, 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 and they're going to flow out of ATP synthase and help generate ATP. This second electron transport chain is making NADPH. This is where our electrons are, and they're still pretty excited. The electrons are not in ATP. We phosphorylate ATP here. The electrons they stay here, and end up in NADPH. We phosphorylate ATP with the help of an enzyme and some proton flow. We're going to take the NADPH and the ATP, and we're going to use it to power the Carbon cycle. You may have learned the dark reaction. We don't call it that because it totally happens in the light. It's just independent of the light. It doesn't need the light to strike it directly, but it won't happen unless the light reaction has occurred. The Carbon cycle occurs in the stroma, and we're not going to worry about the molecules so much. You're not going to have to draw them, but I am going to introduce some reduction oxidation reactions because we like that. So zoom in. This is what we've done. This is one giant thylakoid, and the Carbon cycle is now as part of this process. The Carbon cycle occurs in the stroma. It uses the ATP and NADPH that we made in the light reaction. It uses carbon dioxide, what you're breathing out from the atmosphere. Why are we doing it? We need to reduce carbon dioxide into sugar. We're going to make a sort of a half a sugar called a G3P. This sugar is what the plant uses to run its body. It's what it uses to build cellulose, which is a complex sugar. It's what we eat in plants. It's the whole final bonus product. It's the organic molecule that we're getting by running all of this high energy process. I don't know what turned blue, but we'll go with it. Where it's going on the stroma, we're reducing carbon dioxide into sugar. This is a picture, a very scary looking picture. Same thing I just showed you with the molecules drawn. 50-50 people in the room have taken chemistry and are comfortable, and people who have not taken chemistry. I am not going to ask you to draw these, but if you've taken any chemistry at all, particularly organic, you have learned to count the carbons. I'm going to count the carbons with you. I'm not going to ask you at any point how many carbons, But once you've had organic chem, it makes it so much easier if you have a count the carbon rule for every system you're looking at. Now notice this is not linear, this is a cycle, which means on an exam, I could start you anywhere here and you'd have to go all the way around. But it also means that when I get to the end of this story, I'm going to need to be at the beginning again, because otherwise it wouldn't continue to spin. Now we're really just going to break this wheel into 3 big parts. The carbon fixation part, where we're going to take carbon dioxide and fix it into an organic molecule. The carbon reduction part, that's where we take our organic molecule from part 1 and continue to add more electrons. We're going to reduce it. We're going to add electrons. We're going to add hydrogen ions. We're going to get that from the NADPH we made during the light reaction. We're going to use some ATP to drive these processes. And then once we make some sugar, we're going to have to start up and get back to our beginning molecule again. We're going to have to regenerate our starting molecule so we can do it again and again. We're going to start out with carbon fixation. Remember, it's a cycle. You could start anywhere, but this one makes most sense if I start out with the carbon dioxide molecules coming in. The carbon dioxide molecules are grabbed by an enzyme called Rubisco. Rubisco is one of the most common enzymes in the world. It is really good at grabbing the oxygen side of carbon dioxide. It's also really good at grabbing this molecule RUBP. You know, we go to Rutgers, so it makes it a little bit easier to remember. But this is like RUBP. This is Rubisco. They work together. And Rubisco, the enzyme, grabs RUBP and it sticks the carbon dioxide molecules on it. That's fixation. An RUBP plus carbon dioxide makes PGA. There's a little bit of molecules changing shape and being folded. So for 3, this RUBP is a 1, 2, 3, 4, 5 carbon molecule. We're going to have 3 of them at the same time. Those 3 RUBPs represent 15 carbons. We're going to bind them with 3 molecules of CO2. 15, 16, 17, 18 carbons, 15 plus 3. And that's going to, after a bit of folding with some enzymes and breaking apart, we're going to end up with 6 3 carbon molecules. 6 times 3 is still 18. We haven't lost any carbon here. All we've done is change the molecules and fix the carbon dioxide. We call these 3 carbon molecules here PGA. We're going to add a little bit of ATP. Remember, when we add ATP, we don't just kamehameha it. We phosphorylate with ATP. My ATP, look, it phosphorylates what was my PGA, and it creates a molecule. It looks different, it's shaped different, so we have to change its name. This is 1,3-biphosphoglycerate. Notice I don't have it red here. You don't have to memorize this molecule's name. But this molecule is then quickly reduced by 6 NADPH, its electrons, and its hydrogens into G3P. This is a good question. I left it in here because it's actually part of your textbook. But it wants to know, why is there a positive now on the NADPH? We had NADPH, we built it into our molecule, and then we ended up with NADP positive. What does that positive represent? Where did the electrons go? We reduce PGA into G3P. We actually make 6 molecules of G3P. It's a 3-carbon molecule with a phosphate stuck to it. One of those G3Ps goes on to become sugar. There's another cycle down here that we're not learning. 5 of those G3Ps continue and are regenerated. The little bit of phosphorylation, they're regenerated into our RUBP that we started with. I want you to get the gist of this. So the basic idea. Our 5 molecules will regenerate it into RUBP. RUBP is grabbed by Rubisco, and 3 molecules of carbon dioxide are grabbed by Rubisco. It's an enzyme. It fixes them. It turns inorganic carbon dioxide into organic PGA. PGA is phosphorylated into biphosphoglycerate. Biphosphoglycerate is reduced by NADPH, electrons that we made excited during the light reaction. The hydrogens that we split off of water are built into G3P. 1 G3P becomes sugar. 5 of the G3Ps are phosphorylated. Their shape changes a little bit. They end up as 3 molecules of RUBP, and we do it again. This question here, remember we're hydrolyzing and dehydration synthesizing things, and see if you can come up with some of this. So finally, a final practice question. I think we're going to have to spend some time during review, but I want you to sequence photosynthesis and then just answer the question during the Calvin cycles, where do the electrons move? Do they go from carbon dioxide to sugar, from NADPH to G3P, from ATP to NADPH, or across the thylakoid membrane to the inner membrane? Notice this is A, B, C, D. Only one of these is correct. I really want you to think about where those electrons are flowing. I will see you in review. We'll be doing this Wednesday, Friday of the last week of September. The exam is October 11th. The final lecture on the exam is cell respiration. So this is the second to the last lecture for for exam 1. I will continue on with stuff for exam 2 before we have exam 1. That'll give you time to practice this material a lot. I will see you in cell respiration. I'll see you in review. I hope you had a nice weekend, and I hope you're enjoying this a little bit. Lots of excitement for your excited electrons. All right. Bye-bye. I'll see you soon.